So without all 20 amino acids as a set, good luck. And the key is not just the 20, but the assembly. And the problem is that half produced naturally are left-handed, half are right-handed. And this illustrates the difference. And ironically, only one kind works in life. You must have all left-handed amino acids, otherwise you don't have life. Well, you don't get that when you naturally produce amino acids in the lab. You don't get that. You get half and half. And so this would be toxic to life. And a good example is, there are many examples, but thalidomide, a few years ago when women were taking that for morning sickness, we ended up with a lot of babies that were deformed. Why? Because they used the wrong form of thalidomide, and that's what caused the problem. They tested carefully. They knew that it was okay. But when they had the wrong form, that particular batch was the wrong form, it ended up causing a huge problem with thalidomide toxicity and causing malformation of the fetus. So you have to have, for amino acids, you have to have all left-handed. For sugars, have to be all right-handed. And here you can see chiral compounds, many drugs, sugars, amino acids, DNA, and enzymes. So all of these, left-handed, right-handed, are critical. And Miller believed that one day scientists would discover the self-replicating molecule that triggered the great saga of evolution. It would take off like a rocket. Miller muttered through his clenched teeth. It would be in the nature of something that will make you say, how could you have overlooked this for so long? And everybody will be totally convinced of abiogenesis, that life can come from non-life. What happened? Nothing. This was when he died, I forget, 20 years ago. We're not any closer now than he was. In fact, we're farther away. The more we learn about life, the more we realize how complex it is, and the farther we are away from abiogenesis. So scientists recognize that. Many well-known scientists recognize that. And they realize the complexity problem, which I'll show you briefly. The cell is more complex than a supercomputer. And if you don't believe that, this is a biochemical diagram of the reactions in a cell. And I'm sorry, this is only a small part of the whole diagram, but I couldn't get it in the slide. Besides that, you can't read this one anyways. So I blew part of it up, and there you can read it. There's D-fructose, that's the D form, dextro form, which is the D form. And then there's the other side. So this is this particular chirality of this fructose. Fructose is a sugar. And then you can see all the different relationships that are involved. You have D-sorbitol, xylose isomerase, and so on. You can see all these. And the whole chart, which is 4 feet by 16 feet long, and that is the biochemistry that occurs in your body. And this is the interactome, and what they're trying to show you here is that all of these proteins and small parts have to interact with, and these are all proteins, have to interact with other proteins. And these lines show the interaction. So if you mess up this one, you mess up all of these. And there's not lines here because they're still researching this. When this is done, you're going to have a chart that's probably 10 feet by 20 feet with millions of lines on it. All those relationships are critical. And so therefore, if you have one of these proteins damaged, you end up missing so many things. And that's why so many diseases have so many repercussions based on one small change. And that's why when you diagnose a disease, it could have 20 different symptoms. So it's hard to go back to the cause of all those symptoms. But that's what doctors try to do. And that's because of this interactome.